This morning, it's my great pleasure to introduce our speaker, Bert Tanner from the University of Washington. So Bert, take it away.
Hi guys, how are you? It's
Good.
good to see you. So you guys are all there in one room together with the Summer Institute? Looks like
Those of
there's
us at
maybe,
UCI are, we have a
oh,
couple of
and then
watch
there's a
parties.
few people online too. Okay, this is great. So let me get going here on sharing my screen. So do you guys see something that looks like the Google Drive for the Summer Institute?
Yep, that's what we're seeing
So
right now.
I had put some of the materials in here that if you guys wanted to look at them, just to show you for the workshop 3, there's a bunch of PDFs in here that are basically computational modeling papers that I might be referring to today. And if you wanted to look at them or review anything within that. So just to let you know that some of those resources that I dropped in there.
That's great.
And this should bring up a slide that has an overview of one of those introductory slides from the Integrative Movement Sciences
Yep, we
Introduction
see it.
Workshop. So this is an excellent, I thought, introduction to a lot of the ideas that we've been working with for many years. Some of the people on the call, have thought about all these different organizational scales, and especially how muscle is a really unique system that covers all these different organizational scales that leads to, molecular and cellular processes that manifest into locomotion and movement at the higher order of the organism or even the organ. And then it looks like even some of what's going on with the ecosystem on the very far end. So this is a really unique model system, or an important system for quality of life in terms of how muscle works and what it allows us all to do, both in our lives, but also for many animals across the globe. And so what I'll be talking today about is sort of at this molecular and cellular level, mostly on the idea of like what's going on at the sarcomere and the protein level. So down here at the lower end, do you guys see my mouse cursor? Yeah.
Yes, we
I
can
do.
see it.
And then we'll be talking a little bit about how that affects probably like muscle fiber function and muscle function. We won't be getting up into the larger sort of whole muscle or organismal modeling today. But in general, so I'm up here at Washington State University, and I have been doing computational modeling for a while. And I also do a lot of experimental biophysical experiments, mostly on the cellular level with skinned muscle fibers, both from cardiac and skeletal muscle. And a lot of what we do is trying to better understand, how all of the coupling of the molecular motors leads to muscle contraction at the cellular level with the muscle fiber stuff we do, and then how that could inform movement. And I've shown there some of the important proteins at the bottom, where the primary contractile proteins are the actin, shown in blue along the thin filament, and then myosin, shown in red here, along the thick filament. And I've also illustrated a couple of the thin filament and thick filament regulatory proteins, such as troponin, which is shown in green there, and tropomyosin in pink. Those are important proteins for allowing actin and myosin to interact upon calcium coming into the cell. And we'll talk a little bit about this in more detail further down the way. But then also on the thick filament side, there's other regulatory proteins such as myosin regulatory light chain and myosin binding protein C. So all of this suite of proteins works together to allow for muscles to contract. And over the years, my lab's been fortunate to have pretty decent funding from the American Heart Association, National Science Foundation, and the National Institutes of Health. So that's a little bit of background about my training and what I do. To start to give you an overview of what we'll talk about over this next hour or so is I wanted to introduce some computational models from some of the historical computational models in the muscle field, where we'll start with that with A.V. Hill's muscle model from 1938, and then Hugh Huxley's original actin-myosin cross-bridge model from 1957. Then I've got a few different types of modeling that I'd like to talk about that's related to this class of models called spatially explicit modeling. And as I get more into the detail about this, we'll talk in more detail about some of the ideas within the sarcomere and within the cell that allow muscles to contract in terms of mechanical characteristics of the sarcomeric proteins and the organization of the thick and thin filaments. And then also I'll talk a little bit about some of the kinetics or the cooperativity that allows force to be amplified with a small change in calcium, and some of the important relationships that allow muscle to be a little more effective and efficient in contraction. So to begin with, I just took this slide from a National Institute of Health overview of computational modeling, and the link to it is there at the top. But just in general, computational modeling is this class of science where people bring together computers and they use those computers and the computational modeling to study these complex systems by integrating mathematics, physics, and computer science. And very similar to that first slide we talked about just a moment ago, you see that this particular illustration talks about going all the way from genes to molecular networks to cellular networks and organ networks, and how all that's integrated to allow individual physiological function within a single person, but then that can even lead up to social networks. Very, very similar over multiple scales. This is just a different version of it. And so you see there's a running theme here about many people, how they think about computational modeling as a tool to better understand these integrated scales, because it's very, very difficult to measure all of them all at once. And it's very, very difficult to separate experimentally what might be going on with one particular molecular component that might affect the cell or the organ, because normally a number of these molecular processes are coupled together. So having a computer model, it allows you to separate them out a little more effectively. And so just the most simple idea of computational modeling or modeling in general is to have some sort of a mathematical representation of you think the system is going to respond. And so this is a very, very simple example where you've got a mixture of two chemical components. Say you have substance A and substance B. Those get combined into making something that is substance C. And that arrow there with the K above it is supposed to suggest there's some sort of rate in terms of that K is the rate of which A and B combine to create C. And so how do we describe this with mathematics? Well, this is one example here with a mathematical description where you can use differential equations. And so what this says starting here at the top is this, the derivative of C as a function of time. So this is how C is changing in concentration over time, depends upon the rate K multiplied by the concentrations of A and B. And I think you could understand and think about this. So if there's no A at all, right, then you're not going to have any C being made. And if there's no B at all, you're not going to have any C being made. But if there's some levels of A and B, those then can combine at some rate given by K to make C. And then also complementary, you can write differential equations for how the concentration of A is changing as a function of time. So that's what we have here on the left, where the change in A as a function of time is equal to the change in B's concentration as a function of time. And that happens to be the negative K times the A concentration and B concentration. So I think this makes perfect sense in terms of A and B combining to go forward. Is how the negative version of that would be how the system would go backward, right? Or how you're taking away from A to get to, taking away from A and B to get to C. So that's what these mathematical descriptions are describing. And so then you can solve those equations and see how A and B and C change as a function of time. And one of the important things to point out is you can see here where time is 0 on the left at this particular concentration. Obviously, you have to pick some initial conditions. And so the way these initial conditions were picked is, let's imagine that A, will say this is, obviously A is twice B. So there could be twice the concentration of A compared to B. And that initially there was 0 C. So that's why C is starting from 0. And then you can see how all of this combines with the A and the B are decreasing in time while C increases in time. And so this is actually a mathematical representation of this biomolecular reaction. And this is actually like a very, very simple computational model. And you could then also imagine, like, if you change the concentrations that you're starting with for how much of A there is, and that's what's shown here on the right, is if you start with less A, more A, or some intermediate version of A, then that can drive and change how much C you get, depending on those concentrations. So anyway, does anybody have a general question or anything about the ideas behind the mathematics trying to describe a system? Okay, so this is just a general introduction there. So one of the earliest muscle models that ever existed, and this is actually used quite a bit still today, has to do with what's called this Hill muscle model. And so this person, A.V. Hill, was a very, very famous physiologist. He actually won the Nobel Prize in 1922 for the concept and the idea that as muscle produces work, all of the energy that's used for muscle to produce work doesn't actually go into producing work. There's some sort of efficiency of muscle contraction, and what doesn't get turned into actually doing work comes off as heat production. And so this is a pretty simple idea in terms of efficiency of a motor system, but it also was some very, very elegant experiments that were done about 100 years ago. And then after that, in 1938, he was coming up with some ideas about how to describe the system. And he said, well, if muscle is going to produce some sort of force, it seems to be the way the system is responding is there's a portion of the muscle that is this contractile element, so that contracts. Then there seems to be something that's this series elastic element, that's the SE, that's attached to the muscle. And I think in general, you could think about what this would describe as there's the muscle and then there's the tendon. So you've got the muscle being the contractile element of the motor, and that's attached to the tendon. And that's actually what then would attach to the bone to move the arm or to move the foot or whatnot as force is generated. However, this other passive element has to do with what is a parallel compliance with that contractile element and that series element. And that might describe sort of the connective tissue that's in parallel to the muscle and the tendon perhaps. So that's what these different systems represent. And you can change elements of this to describe and show how force is produced in a lot of systems. And this is a simple idea, but it doesn't have any like actual details about what the actin and myosin cross bridges are doing. This is more of a larger scale Hill model. But the ideas that they're trying to model within there is this passive element. You know, as you increase the muscle length, you'll see that there's a passive element that as you get to longer and longer lengths starts to come up in blue and produce more and more forces. And then there's this active contraction. So as the muscle is contracting, that's what's shown here in red as a function of muscle length. As you get some sort of a plateau or a maximum contraction for a muscle twitch, and then at longer and longer muscle lengths, that starts to go down. And if you combine the active and passive, you get something that sort of looks like this green line. And this model on the left can have characteristics that try to describe these different systems. And that's the idea of a hill model. At a more detailed version was this idea from from A.F. Huxley. I made a typo earlier where I said it was Hugh Huxley, which is actually A.F.'s cousin. They were a pretty skilled family of scientists. And A.F. Huxley here made this crossbridge model in 1957. And a lot of what I'm going to be talking about today sort of builds off this idea of this crossbridge model. And so one of the portions of this paper, you see this paper was about 70 pages long, you know, just a little afternoon of work for A.F. Huxley back then. And he had this one section, section 4, where he puts together this entire hypothesis of this mechanism for contraction. And I've taken a few snippets out of that to try to describe or show what these early ideas were about how actin and myosin might bind, and this concept of some sort of position dependent or spring-like dependent response of the actin and myosin system. And so A.F. Huxley here described or showed this diagram for which tension is generated, where there's the thick filament of myosin and the thin filament of actin. And the concept is that, you know, myosin, as you contract, is going to slide this way and actin is going to go that way. But what's going to make them move is that interaction here between the myosin motor, the cross bridge binding to some actin site. And depending on how far apart those are, and depending on how the actin myosin are swinging past each other, there's going to be different probabilities of how they bind and how they might produce force. And here you start to see this spring element where they're trying to have some sort of a position dependence of this rate. And so if we were to think about some of these rates, they put together this idea that the attachment rate for actin and myosin binding is this rate that's a rate constant called F. But if you look at the bottom over here, you can see that F changes as a function of position, depending on the distance or the difference between that A and that M that we were showing just previously, and also up in this top corner. So this is position, this X is position on the bottom, and the rate constant is vertical here. So you see that F is this dashed line that, depending on how aligned actin and myosin might be, you get this increase in force or this increase in attachment rate until all of a sudden there's some decrease where it goes back down to 0, where it's just too far away to bind. And then similarly, you get this detachment rate, and that's shown by actin and myosin detaching, and that's this rate constant G. And so here you see there's sort of some sort of a smaller slope with G here. But if you have actin and myosin go at all the opposite way, so after maybe it's through its power stroke as an idea, you get a very, very high rate of G, and that's over here at the negatively strained or negative X values, you have a high detachment rate. And so A.F. Huxley went forward just to describe that, you know, if you have actin and myosin sliding past each other, what you might see in terms of the probabilities of binding, if you're not moving at all, you'd have some sort of interaction here where you'd get actin and myosin binding at maybe, say, 80% as an example is what they took here. But as you increase the rate at which they're coming past each other, so this is your velocity is going to 10% of Vmax, 20% of Vmax, or like the maximum velocity that you can be sliding past each other. Well, that's going to change the shape of this in terms of the faster and faster that actin and myosin are going past each other, you can see that the relative height of this binding curve decreases and decreases and decreases. And at the same time, it sort of spreads out. And the reason that sort of spreads out is because of how you're stretching that bond, or that actin and myosin acting like a spring event to generate force as the filaments are sliding past each other. So this was just one idea where he was trying to explain some of the force velocity relationship as a function of position for how actin and myosin were bound. And recently wrote a little review that has to do with introducing some of the spatially explicit models, which I'm going to talk to you more about in a minute. But most simply, right, you can imagine, and the modeling that I'm going to be showing you is effectively like a model of the half sarcomere. And you can imagine that whatever goes on for activating this half sarcomere model has to do with calcium coming into the cell. A lot of what I've worried about over the years has to do with changes in sarcomere length and how the filaments are overlapping and lining up. And then that leads to some predictions about how muscle force is produced, how fast the cross bridges might be going through the cross bridge cycle. It has to do with how calcium activates the filaments. But that's the most simple higher level. But if you get down into the details about it, somewhere along there, I have to make equations that describe the geometry, such as how the thick and thin filaments are arranged, or how the regulatory proteins lay along the thin filament. Or there's these ideas of mechanics. So I have to make equations for how stiff those filaments are, and maybe how that would affect how actin and myosin interact, or how force is generated to propagate through the sarcomere. And then again, there's the idea of the kinetics in terms of how fast actin and myosin might be interacting and things like that for attachment and detachment. So this has a lot of detail behind it that I'm not really going to talk about today, but you can see that qualitatively, there's nothing very much different from that introductory biomechanical reaction we talked about a few slides ago, where you're trying to put forward some equations to describe the system the same way we put forward some equations to describe how A and B come together to make C. So those are some of the general ideas behind computational modeling. Before I go into more detail here and starting to jump into some of this spatially explicit modeling, where we're actually trying to model the half sarcomere and characteristics of the half sarcomere, does anybody have any questions or anything we want to discuss? Not yet. We can come back in the end. Am I overwhelming everyone?
No, I think we're following along okay here. Thanks.
Okay, cool. So as I mentioned, today I'm going to talk a little bit about those spatial geometry, mechanics, and kinetics that might underlie how muscle functions. And so a lot of what I'm showing you has to do with some stuff that I published, a while back, but it's some of the most introductory stuff about trying to separate these different characteristics of muscle that might come together to produce contraction. And so this is sort of a cross-sectional view or an end-on view of, if you were looking down the myofilament lattice, you can already see there's a few things to keep track of in terms of where actin and myosin are aligned and where they might line up and who they're pointing to interact with each other, where the myosin is in red and the actin is in blue, and then the regulatory proteins along the thin filaments are sort of shown there in yellow and green. And, basically we're trying to say what goes on with this system of molecular motors and this network of molecular motors, and how does that underlie muscle contraction? And so generally we have muscle converting chemical energy into mechanical energy, and there's a few characteristics that we have to keep track of related to that. And so if we think a little bit about the geometry of muscle, we've already introduced this just a little bit. So, if you have a whole muscle, then that pairs down into a fascicle, and then ultimately that pairs down into a myofibril. And you see the striations here of that myofibril that come from the sarcomeres, where you get this overlapping interdigitated filaments of the thick and the thin filaments, and that leads to the striations you see in the sarcomere in striated muscle. And striated muscle most generally is skeletal and cardiac muscle. And then you see down here, I've introduced, again, this is the same picture, just a little different version of the myosin with the cross-bridge heads and how they're binding to actin and some of the regulatory proteins along the thin filaments. And so this is a calcium activated process down here, which I'm not going to talk a whole lot about in the beginning, but we'll come back to in the end. And one thing that we're trying to compare, or one thing I'll compare in a minute, is, well, what are the effects of all these interactions from that thick and thin filament lattice and how they're organized there where the thin filaments are in blue and the thick filaments are in red, because they're not just lined up with each other on the page. There's actually like a specific ratio of every thick filament points to 6 thin filaments, and every thin filament has 3 different thick filaments pointing at it to potential have those interactions there. And so I'll compare a little bit of our predictions with some measurements to try to validate the model, because it's always helpful if you can use experiments to test your predictions. And if they line up, it helps you think you might have the right idea of what's going on. And so the events, or the kinetic events, that I put into this particular model have to do with some aspects of thin filament activation, sort of shown here on the left, and then aspects of the cross-bridge cycling, or the cross-bridge model. And so if we think of going from this first state where there's no calcium in the system to then calcium binding to troponin, that's this first transition. And then the next transition is tropomyosin moving out of the way. And as tropomyosin moves out of the way, you can see that then that might allow for the cross bridges to cycle. And when the cross bridge is detached, it spends most of its time in this position where it has the ATP hydrolysis products, ADP and phosphate, bound to the cross bridge head. And then that goes to then bind with actin. And then as phosphate is released, you get this power stroke, or the force generating event. And that translates actin and myosin past each other as the cross bridge goes through the power stroke and releases phosphate. And then ADP comes off and ATP comes on. And when ATP comes on, that's when you dissociate. So that's sort of the background of the kinetic events in the model. So that's sort of the end of the introduction there. Do you guys have any questions we should go over before we get into some of the modeling results?
Any questions here, team? We're good here.
Okay, cool.
But can I ask a brief question?
Sure, Madhu.
It will. It doesn't have to be now, but at some point, I would love for you to annotate which of that molecular picture has active debates versus what you would consider as resolved.
The previous picture?
Yeah, in particular, yeah.
Okay.
But even things like the cross-sectional organization, right? I'm beginning to learn from Natalie, for example, that some of these giant sarcomeres can vary quite a lot in the cross-sectional organization and so on.
Okay. The organization in vertebrate system doesn't change that much. The organization of the thick filament structure and invertebrate systems is very diverse. That's a real easy summary of that one particular answer. And the thick filaments seem to be, the length of the thick filament seems to be pretty constant in vertebrates, but there's definitely some diversity amongst the thin filaments, and that can vary with muscle type, and it can also vary with different organisms much, much more. And so those briefly are some of the structural things that are a little more diverse compared to a little more consistent.
Okay, well, yeah, it's mostly I want to hear your take on it, because you've thought and looked at every piece of this in such a detailed manner over the years. It would be really helpful to get your view on, including the kinetics picture. I know there are debates about specific sequences of events, like the phosphate release versus the power stroke, which you think affect the way you envision the spatially explicit dynamics working. I mean, there could be debates, but they may not have, in your experience, much bearing on what the predictions of your spatially explicit models are.
Okay, well, that sounds cool. We certainly could discuss that for many, many hours.
Sure. Okay.
So yeah, cool. So I've got a few examples here of trying to test some of these model predictions and trying to understand how some of these characteristics might influence muscle contraction. And so the first one has to do with thinking about, well, what are the mechanical characteristics of these filaments? And there was some really nice papers that came out in the late 90s that suggested that as muscle produces forces, it actually sort of stretches the filaments. And the filaments have a little bit of flexibility to them and they can extend. And what might the consequences of that be? Because before, a lot of these models assumed that the filaments were effectively completely stiff and didn't have any extensibility or any flexibility. And so, as I mentioned, there was this Huxley model where they were looking at the different characteristics of the actin and myosin interaction, which we sort of introduced a little bit before, and that was just looking at one myosin. And then my PhD advisor in 1998 published a paper where they said, well, what goes on if we have two filaments? And if those filaments are allowed to extend or be a little bit flexible, what are the consequences of that for this half sarcomere model of two filaments? And then the model that I made essentially has 4 thin filaments and 4 thick filaments and 8 thin filaments. And if you were to imagine this little like dashed line that I've drawn around the cross section of the sarcomere, I made it in a way that basically one side of the boundary wraps back onto the other side to try to take away these like edge effects, or simulating a little subsection of infinite lattice space. So that's a little modeling trick that I got from some computational fluid dynamics, where they have these sort of periodic or toroidal boundary conditions. And that allows us to sort of have a tractable number of filaments without any of these strange edge effects that you can get if they didn't have those same boundary conditions wrapping back onto each other. And so I've got a couple experiments that I've done, and then also some of where a lab in Seattle where I tested some of the predictions from what they had done. Right. And so one of these assumptions that came from this early model of the Huxley was that myosins are flexible or have some sort of a spring-like behavior, a spring-like elasticity. There's that position dependence of how actin and myosin bind. So depending on how they bind has to do with how far away the binding sites are from the crossbridge head. And then this was sort of a simplified kinetic in 1957, where it was either bound or not bound. But I showed you that there's a few more steps in the cycle that I had measured. And originally, Huxley had done this with differential equations, and that assumed a well-mixed population of crossbridges. So where everything is happening is described by one crossbridge, because that would be like the average behavior of the crossbridges in the system. But if we're going to be keeping track of all the crossbridges in this network, it's very, very difficult to describe that with partial differential equations, and is impossible to describe with ordinary differential equations. And so we did this sort of stochastic approach. And one of the ideas that came out of that modeling from the Daniel 1998 paper was that some of this extensibility or flexibility of the filaments might actually lead to increased crossbridge binding and increased crossbridge cycling because of the filaments being a little bit flexible. And the general idea with that could be like, say this crossbridge binds where it goes from red to pink, and then it generates its crossbridge force, right? Well, you're kind of going to get it to stretch out down here, and that'll push the thin filament past the thick filament, and that might align better. So if we go back there, before you have the power stroke taking place and stretching out the filament, now you get better alignment of your neighboring crossbridge and actin sites, and that increases the probability of binding at this particular neighbor, for example. And so that might lead to greater crossbridge binding. And one of the questions I wanted to know is like, well, what's the difference between two filaments and if you have these multiple filament interactions, okay? And so just to sort of predict how that might occur there, this is the little concept, is that the flexibility of the crossbridge lattice allows more crossbridges to bind and maybe allows for more force production to occur. And there's a lot of details in here, that we're kind of gonna skip over. But one of the ideas that came out of that paper, from that Daniel 1998 paper, is if you think about how tension might change as a function of, this is an example plot of changing the thin filament stiffness or the crossbridge stiffness, you see that you get this range of tension values, or these range of force values that can be generated, and that can affect your maximum tension depending on the stiffness of the system or the flexibility of the system. And it turns out, for our best estimates of what the crossbridges stiffness is and the thin filament stiffnesses are, it's somewhere close to the maximum, but not the maximum. That's what's shown here by the star. And so this would suggest that some of this flexibility makes it so crossbridges are affecting each other when they bind. And as I mentioned, I wanted to compare the two filaments to the multiple filaments. And so what I was able to do was to use my model and take a bunch of the interactions away to then compare amongst, I thought I had a slide right here showing this. I'm sorry, you guys. So anyway, we've introduced a lot of this background. You can see that there's all these springs that describe the system for the thick and the thin filament and the cross bridges. We were able to compare using that kinetic system that I showed you before. You can use all those equations to describe how this might be. And then we were able to look at the ratio of the filaments here by comparing just interaction of one thin filament and one thick filament pair here, similar to that 1998 paper from the Tom Daniel, compared to the whole model here. And the first thing we saw comparing between those two models, if we have this multiple filament model compared to the two filament model, is you get a lot more force. But the reason this particular metric of force is so much greater is because you have so much more interactions in the multiple filament model. So if you were to normalize that to the relative number of myosin interactions, right? So now we've scaled with the number of possible interactions that are occurring. You see that that difference is not quite as great, although there's still a difference where you get more force per myosin in the multiple filament model than that two filament model. And this is very interesting for us to initially observe. And it turns out that the idea of the measured piconewtons per myosin that are generated, the predictions from the multiple filament model were much closer to some of what was measured with some single molecule actin and myosin crossbridge measurements that came out in the mid to early 2000s. And so that was sort of an interesting initial finding. Another couple things that came out of comparing the multiple filament model, the two filament model, was that you got more binding. You got greater crossbridge binding, and you also had faster or more efficient ATP turnover in terms of how much ATP was used to go through the crossbridge cycle per second. And all the predictions of what came out of that multiple filament model seemed to align more closely to what was experimental measurements compared to the two filament model. And I think the reason that this occurs, that the multiple filament predictions are more consistent with experiments, I think the reason is because you have more of these interactions, that you get more of that realignment or the more stretching of the cross bridges and the filaments with more great cross bridge binding due to the coordinated recruitment and turnover that comes from the geometry of the sarcomere and the multiple filament interactions that you're missing in that two filament model. So one of the ideas to take away with here is that this coupled cross bridge binding depends upon how many motors are able to generate force and how those motors are lined up to bind with the actin and myosin events. So the organization of the filaments also seems to matter. So that was one idea. Do you guys have any other questions on that? Or does that make sense qualitatively about how the geometry and the flexibility of the system might affect how the motor proteins behave?
Yeah, we're nodding our heads here.
Okay, cool. Awesome.
Gonna keep this unmuted.
Yeah, just really quick. So, is there a higher proportion of cross bridges attached in the multi-filament model at any given point in time, or?
There are, Kisa, this was it right here. It's like a roughly from, with all of the stiffnesses being equivalent, or, you see about 5 times more cross bridge binding.
So it's the number of cross bridges bound and not the force per cross bridge that's changing in your model.
Well, the force per cross bridge also changes. That's what's shown here on the left.
Okay,
So the
good.
force that's generated per cross bridge is greater. The number of cross bridges that bind that are greater. And it seems to be a much more dynamic network in terms of many more interactions and going through the cross bridge cycle to generate force, more cross bridge binding and more turnover as well.
Right, so you have a higher ATP cost on the third column. Right, got it.
Thank you.
No problem. Thanks for reiterating those details.
Do you have a follow-up question?
Yeah.
Is the increased cross-bridge force due to a difference in its length, like its delta X?
How do you get more force per cross bridge? Is it a torque to force conversion like geometry, or is it something about stretch of elastic elements between the cross bridges and the attachment?
I think you're getting more filament realignment and more cross bridge binding, which leads to greater recruitment and greater force production in the whole sarcomere system. I paused on answering your question on if the distortion of the cross bridges is different. I've never looked into that, but you could imagine that maybe on average there was some change in the average position or distortion that would lead to greater force borne across the cross bridge. But I think it's more
Is like
of the
amplified
dynamic realignment, because the underlying mathematics for the kinetics and the actin myosin binding in terms of the probabilities of binding is a function of how the binding site and the head are. The mathematics is identically the same. But that's an interesting question that I don't actually have the answer to in terms of how the cross bridges are strained differently.
Yeah,
It could also just be a torque to force conversion, like moment arms of the ratchets. Is that a possibility?
Well, there's no difference in the structure of them in terms of the cross bridge itself. But
can the distance between the actin and myosin change in the
they're
spatially explicit model?
A different angle when they're bound.
they
Across
can
the distance.
change, they can change like sliding past each other, but they can't change like vertically, like how far apart their
okay.
spacing is.
So they can't go this change.
So
Yeah, okay.
But I mean, Simon talks about how they do get closer together.
You're saying the moment arm that matters is constant because it's the vertical
Yeah.
height between the, okay.
And
Got
so
it.
there's a series of models that has followed this, and I think Simon and Travis Toon have started working with a version of this modeling idea where they allow the like what would be the radial distance or the distance between the thick and thin filaments to change depending on forces and how the cross bridges goes through that sort of torque or ratchet event you guys were talking about, that is not modeled or possible in the particular model that I'm showing you.
Okay, so I have another question. We kind of want to ignore everything you said in our model. Does it look like an amplification, or does it fundamentally change the dynamics of the force generation?
It changes the dynamics of the force generation. And I think the reason that occurs is because you have much more realignment and binding in the multiple filament system compared to the two filament system.
Right.
Madhu has his hand raised.
But two quick questions. Now, one is the two filament model still uses the same lattice spacing of the motors as the multi-filament model, right? So is there a variant, meaning an equivalent two filament model where I can increase the density or some such by thinking about averaging across the cross section? That's question number one.
I don't understand the second portion of your question. The first portion, the answer is yes. The second portion,
So I can imagine like in any sort of intermediate step of developing a continuum theory, I can start averaging across things. And one average I can compute is a cross-sectional average and ask what would be an equivalent to filament model where the exact details of the spacing parameters, for example, have to be changed, or exact details of the Meissen cycling have to be changed. But it's then a representative two filament picture where I've averaged across the entire cross section. Have you or do you know of others who've tried to construct such coarse graining approaches?
I have not. I have not. And it's not like off the top of my head that I know people who've tried to sort of do this potential coarse grained approach or sort of intermediate modeling moving towards continuum theory that you're suggesting. I don't think I've heard or know of anybody trying to do that.
Okay, a slight minor second question. How do I think about thermal fluctuations in this context? For example, when you say that the stretch of the filament allows better realignment, there is some local strain energy in the filament itself. How does that compare to just KT at that scale? So in other words, do thermal fluctuations regularize or smooth some of these extra alignment you get, and therefore the differences become smaller?
Well, so we tried to tie all of what underlies the kinetics together related to potential thermal fluctuations of the probability of binding of the actin and myosin organization in terms of how that would disturb or distort the crossbridge head as you were to either change temperature or change stiffness based on some thermal energy in the system. But in terms of thermal fluctuations affecting local strain, it more affects the crossbridge kinetics, nothing actually to do explicitly with the local strain.
Well, what I have in mind is the configuration of my myosin motor is not a deterministic thing. There's the some of it is abstracted away in these rate functions, but I'm asking that crossbridge itself is fluctuating, and it's fluctuating due to KT fluctuations. So if I look at the additional strain that you're computing in the filament that causes the alignment, how does that strain energy compare to KT, where T is now body temperature for it? Am I of the order of KT? Is the strain energy, elastic strain energy, many-fold, like many orders of magnitude higher than KT so that I can ignore KT? Or if it's less than or of the order of KT, I cannot ignore these configurational fluctuations.
I don't know. I might have to share with you maybe this is off the top of my head. I don't exactly know the answer to your question in terms of the orders of KT. Like how many KT is ATP hydrolysis?
ATP is a good question. I think it's of the order of
Like,
1.
so whatever it is, it's like 20% of ATP hydrolysis for however many KT that would be.
Okay, thank you. That I can do the arithmetic, and if I will still come and talk to you more about this, but that helps.
Okay, we have a few other hands out there, it looks like, yeah?
Yeah, we have a question here. If you want to go ahead.
Yep, I was just wondering, since we're assuming that the filaments are pliable? Is there some range that we're operating within? Like, is it within an elastic range, or are you just assuming that, they're not gonna vary too much between each other? The filament length?
So within any particular simulation, we set the stiffness characteristic or the elastic characteristic of the thin filament, the thick filament, and the cross bridge. But we've done a number of simulations where we've varied those relative stiffness values to try to understand how sensitive the model is to one of those particular values versus another one. And so within one simulation, they don't vary, but you can change them. So you can ask, if you change it 10%, what's the effect? If you change it by a factor of 2, what's the effect? And so I don't have any of those data in this particular presentation, but those simulations are within some of the papers that I uploaded. There'd be the PLOS computational biology papers from 2007 and a little bit from 2012.
Thanks.
Thank you.
And I could open those right now if you wanted me to show you, if we were interested in that. Or if this is like, it's better to have a good discussion than to show you a whole bunch of data.
Everyone else.
We got 4 minutes, we can do it.
I think, well, if you have more to cover,
Well, I mean, it just depends what we want to cover. I can show you, I have a couple more examples of some things we've done, but it's nice to have a dialogue than just show you what we've done.
I think we can look at the papers later.
Okay.
Yeah, we'd like to see your examples.
Okay. So another thing we wanted to say or ask is, as you activate the thin filament, as calcium binds and you activate the thin filament, there might be some coordinated coupling of activation along the thin filament. And we were a little curious as to how this might compare to the rules of activation physiologically. And so if you think about the organization of a thin filament, there's sort of this idea called a structural regulatory unit. And that's shown here on the left, where you have one troponin, one tropomyosin, and that one tropomyosin covers 7 actins. So the distance of it. And so if you were to imagine just one unit activates, you'd sort of get 7 actins per calcium binding. And I've shown 3 different regulatory units here, but only the middle one is activated with calcium binding. And so there was some experiments that were done where you can actually change out the troponins so they can't bind calcium. And so you can change your density of your functional troponins by the ratio of your like native troponin to how many you've knocked out. And that's sort of this rho Tn is how many, the fraction of troponin that can be activated. Okay. And if you look at the data and you were to change this whole troponin from like a very, very low level of what can be activated to 100%, and this is at maximum activation of force in a skinned skeletal muscle fiber, you see there's sort of this increase that's sort of a little bit of a nonlinear response compared to like the unity line shown in black. You get this sort of increase that is the greatest divergence is sort of somewhere in the middle from 40 to 60% activation. You get more force than you would imagine compared to just coming back to the 1-to-1 line at 100%. And so this would sort of suggest that there's some sort of coordination where there's a way that you're getting more than just your like 7 actins being activated by 1 calcium binding. It might be that maybe you get a little bit more because these troponins on the end, troponin actually overlaps each other. And maybe that one tropomyosin might effectively roll or move its neighboring tropomyosin just a little bit. So you get more than just 7 actins available with calcium binding. And that is this concept maybe called a functional unit of activation rather than a structural unit of activation. And that might lead to greater cross-bridge binding. And so these data that are in blue and orange are actual experimental data. The black here is the model predictions. So this is if you just got 7 actins activated. You see it pretty much follows this 1-to-1 relationship for maximum force as a function of activatable troponin. But if you allow that span to be roughly 7, then you can see that this sort of follows the line pretty well compared to the exact data. And so that would suggest that maybe functionally calcium activates about 11 actins instead of 7 actins. So that functional regulatory unit is a little bit bigger than your structural regulatory unit. And that might be one way that, you know, another component of thin filament geometry, or the way that the proteins are organized along the thin filament, that helps to increase the thin filament activation and maybe lead to also greater cross-bridge activation as well, or cross-bridge binding as well. So the spatial coupling along the thin filament might also be an important component. And so between the two things I've just shown you, it suggests that both the cross-bridge or the filament geometry, as well as the thin filament regulatory protein geometry or activation, those seem to also be coupled together. So that was one interesting idea that came out of some of this modeling. And you see that if you look at what goes on with the relative force as a function of the calcium level, where PCA9 is low calcium and PCA4 is high calcium, with this functional regulatory unit span, we see this force PCA relationship, which is pretty characteristic of what goes on, but there's some differences in terms of the slope that we could go on and talk about. Although I know this has been going for a while, that was the last little section, is if you were to imagine what goes on with these ideas of how force is actually developed in a skeletal muscle, you see that the steepness of the slope shown in blue, or the steepness of this force calcium relationship, is much larger than what we were predicting with the models that I had initially shown you just a moment ago. And so this kind of brought us back to wondering, well, are there additional mechanisms of what's called kinetic cooperativity, or different allosteric mechanisms that underlie this cooperative response? And physiologically, this is an important response in muscle, that for a very small change in calcium concentration, you get a large amplification of the force that's produced. And that allows for your muscles to be able to increase force production without having to shuttle as much calcium in and out of the cell, the more steep or the more sensitive this force calcium relationship is. And we thought of a few different ways that this might affect the kinetics, and we tried to model these different ways this might affect the kinetics. And so there were a couple ideas, like I've shown you spatially already with the spatial activation of the thin filaments with the functional units. But it could be that as calcium binds to one of the functional units, that might have some sort of a feedback effect on its neighboring regulatory units, where it could maybe increase calcium binding, or it could increase activation probabilities. So that's what I'm trying to show here with the orange ones feeding back on this forward rate. Similarly, you can imagine if a cross bridge were to bind and stabilize activation of the thin filament, that could increase calcium binding or activation at a neighbor. And that's also what I'm trying to show here with the magenta. And then intrinsically, there's a little bit of that cross-bridge cooperativity that occurs in the model due to the compliance, which we've talked about a little bit before. But because it's a model, you could actually say, oh, well, what if we were to change how that increases the probabilities of cross-bridge binding if neighboring cross-bridges were bound? And so all this stuff we could try to test out and simulate a number of these different events. And so I just tried to compare the model sensitivity to these different components, you know, depending on these different ideas, where if calcium were binding here, it might increase activation at the neighbors. Those are the functional units of cooperativity by feeding back on these particular rate constants. And then similarly, like I tried to measure, if you've got binding of the cross bridges, that could increase the probability of thin filament activation. And we did a whole lot of different components of measuring all this and putting all this together, where again, in orange and blue are measurements from skeletal muscles. And depending on the combinations of what we put forward, we were able to put a specific combination that seems to simulate or emulate a lot of what was measured, depending on how you combine those relative portions of allosteric cooperativity and how that might affect thin filament activation kinetics. And there was a lot of different simulations we tried to test here, but it required you to combine all the different forms of the cooperativity. Not one of them was individually sufficient by itself. And so that was just one additional idea, is that, you know, a lot of what's being coupled has to do with how the system has its mechanical characteristics, its spatial organization, but also how some of the kinetics might affect the neighboring regulatory units as well, to actually start to be able to simulate force production in a skeletal muscle a little more. I don't know if accurately is the right word, but to align the model predictions and the experiments a little better. And so in conclusion, we saw that contraction seems to be important to couple all these interactions for cooperative protein interactions. We see that because of the additional filament interactions with the multiple filament model, that seems to coordinate cross-bridge binding and increase thin filament activation. And it seems to be that, you know, what goes on with cross-bridge binding, like some cross-bridge binding begets more cross-bridge binding. So that population effect might actually scale more greatly if we were to scale the models up, or to think about, you know, one myosin motor versus millions and millions actually that occur in a muscle. So some of this might even be amplified in a real physiological system. And so one of the real shortcomings of everything I've shown you is all this was isometric contraction. But as we know, and I know a lot of people have talked about within the grant that was written, that a number of us have worked on, is that muscle contraction is a dynamic system. And there's a lot of different examples of this, trying to get into better understanding whether this is oscillatory contraction in insect flight muscle, or within a heartbeat, or force velocity relationships in vertebrate systems. There's a lot of additional simulations and measurements that can be done to better understand the dynamic system. And just to follow up, you know, this I showed you before, and as you see, a lot of these ideas are consistent with what I showed you earlier, but we were sort of modeling at the lower end of this. The integrative theme 2 within this grant can expand a number of these ideas, where we were talking a little bit today about down here at the spatially explicit models, but there's a lot more different types of models moving up the organizational scale as well. And one more recently developed tool that's actually an open source tool was out of Ken Campbell's lab at Kentucky. And this is this FiberSim model, which is very similar to the types of models that I talked about today, but it's an open source model that might be something people are interested in playing around with, or is more accessible than some of these other tools. And so those were just some other resources. And with that, I'd be happy to talk about whatever we want to talk about going forward, but that was all of the examples that I had to show you.
Thank you. Bert, does anybody want to start off with any questions? I can kick off a question if people are still thinking. The results that you presented are really intriguing, but it looks like it's very computationally intensive to run these kinds of molecular level simulations. And so I was wondering if you have thoughts on, or could speak a little bit about how you would abstract some of the findings to be able to integrate them at the higher level models in ways that are not too computationally intensive?
So some of the stuff that I've done is definitely more computationally intensive than some other things.
Mhm.
This particular fiber sim model, Monica, is very fast and much less computationally intensive.
Mhm.
And that's because it makes some generalized assumptions and how it keeps track of some of the kinetics are a little bit simpler. Some of the solution algorithms for how they're doing all their balances of forces are a little bit different in terms of using an optimization algorithm compared to linear algebra. And one of the other things that has made this tool a potential tool I'm looking into quite a bit to learn more about that might be more computationally tractable is a lot of what it is being written in is Python and C++. So those run quite a bit faster than some of the other stuff that I've written. Most of my modeling was written in MATLAB, which has got some drawbacks. The other thing I've thought about outside of that with the fiber sim is that I think there's ways that you could probably try to integrate some waypoints along the way where you could take like snapshots of what might the system look like rather than having to coordinate with a higher level or more broader organizational scale system at like certain time points along the way, like in terms of if you were to take a snippet or a snapshot of like what is going on at the molecular level, rather than simulating every millisecond or 1/10 of a millisecond, maybe we take a snapshot every 20 milliseconds or every that you'd integrate with another model or something like that. So have a little bit of the back and forth that is a little less intensive for how they're all coordinated for the temporal communication or informing the dynamics of the other model might be one idea.
Okay.
I have a question. If nobody else does. So, Bert, I know that we've talked a lot about this and thought about this, but I thought it might be interesting for the whole group, to think about, as we move forward and we have, like 5.5 years left on our 6-year grant of, specific ways that, you and I and others might, and Madhu and others might think about, how to add to the work that you've done. And I was, one of the sort of the bigger ideas out there, that's fairly new is the idea that, not just the thin filament is activated, but also the thick filament. And it seems like there are some recent studies out there that suggest that Titan might be playing a role in activation of the thick filament. And it's very much wrapped up in the compliance of the thick filament and how that affects, the mechanics of the system. So I don't know if you just have some ideas you'd like to share with the group about maybe, is that a direction that, would be interesting to move forward in? And are there other ones?
I think it's a really important direction, and in terms of if you guys understand or don't understand, so if we were to just look at some of what I put on that Google Drive, for example, some of what Kisa might be talking about here could be related to this idea where you've got this is a little bit of a different characterization of the kinetics. But as you see here, what this particular paper was talking about was the blue is showing what's going on with the thin filament, where you've got an inactivated thin filament, an activated thin filament that can allow for cross bridge binding. And then if you think from the cross bridge side, Kisa was talking about this idea that there's some component where you've got this off state or super relaxed state, and that goes to this disordered relaxed state where your cross bridges are now available to bind with actin, whereas in this super relaxed state or off state, they can't bind. But here in the disordered relaxed state, they could bind. And then that leads to this force generating event. So you see the way that they modeled this transition, Kisa, was I believe that their K1 or their recruitment rate from the SRX to the DRX had to do with how much force was being generated in the sarcomere. So that was effectively a force dependent recruitment. It wasn't necessarily a strain dependent recruitment, which I think if you're modeling an entire system like an elastic system, there's a real close tie with that strain dependent and that force dependent recruitment, but they didn't do it with local strains. They did it with like the overall sarcomere force production and how that would affect the system overall. So that is one kind of idea of something that has been done that I think various ways could be improved upon, but also various ways could be implemented within the type of models or class of models I showed you today, or it could also be implemented with some of what's already available with that FiberSim model. The Titan question, I know a little bit less about related to the FiberSim system, but I mean, I've got Titan in some of the models that I've generated. And one of the ideas that I didn't upload a paper about, but I think might be important to think about is there's differences in how you might model Titan, whether it's a linear stiffness or a nonlinear stiffness as you stretch out the system. And that would have a big impact potentially on stretching out the filaments in terms of any of that strain dependent recruitment at the local level for the off to on transition and improving or testing regulation as a function of sarcomere length. I think that has some very important implications we could consider. So I'm not sure if I'm answering your questions. I've thought a lot about this. I just haven't gotten to being able to do it yet. One of the drawbacks of that, but I think one of the opportunities we have is there's a bunch of interested and eager folks to try to work on some of this across scale over these next few years. And one of the things that's sort of been languishing or slowing down my modeling progress is I haven't had any students be interested in computational work in quite a while. So that falls back onto me doing all the modeling up here, which doesn't happen very frequently.
Yeah, so this model does not have like a activatable Titan, but it does have a series elastic element that is linear. I use linear ones, but you could activate it.
So you guys have been a little bit familiar with this?
Yeah, we have our models based on, and we were just up there a couple months ago, and our crossbridge model has a super relaxed state also, but it also has this series elasticity, and we have found that when we try to fit the skin fiber data, that parameter needs to be changed. Like it's not really constant across what activation levels. So that's been some of our model fitting is driving us crazy.
So your screen says Kristen Jakubowski.
Sorry, I'm Lena Ting. That's
Okay.
Kristen. There's Greg Sawicki. He's done the modeling.
Hi guys, it's good to see you. I didn't realize who was talking. Like now that I know who you are, I'm sure you know a lot about this, given all the work you've done with Ken and some other things like that, and all the other modeling all you all do. So thank you. I couldn't see on my screen who people were.
No, that's fine. We're in a big group, so.
Surabhi, who's further back on the table and you probably can't see, is the one leading the modeling work in the groups. And so it would be, maybe she can visit you, Bert, at some point. I think that would be good.
Yeah, I'd be happy to talk more with anybody on any of these ideas. And this is really cool to even be able to just be sharing some of this or talking to people with getting things going on the summer workshop for how quickly all this came together. I think there's a lot of neat projects that people are reviewing or going over for the summertime that hopefully we can keep rolling on throughout the academic year.
Yeah, and we should absolutely send people over across our different institutes to try to collaboratively train a graduate student to work on this with Bert, right? So because that's part of our goal is to help train up people to work across our network. So we know of some good graduate students who might be interested.
Yeah.
Yeah, and just to quickly say too, that we're very soon going to be putting out an advertisement for a postdoc between Bert and me and Madhu for Core 1. So if you know of anybody who's at that stage who might be interested in the position, please have them contact one of us. So we'll be looking to hire within the next 6 months. And I also just wanted to say, in terms of like data sharing and things like that, that we've been doing some system identification on intact mouse muscles to look at how, the stiffness and damping constants of muscle are changing with length and activation. And I think we have some really, pretty model neutral estimates of, effects of length and activation on like the series compliance. And if that would be useful to anybody to, give you ideas about, how to potentially parameterize your models, like that's totally a resource that I hope to share soon, and the data itself will be available so people can do their own analysis.
Any other comments or questions?
I really like the introduction. I just want to throw that in there.
Oh, sure. Yeah. So I appreciate that. I thought it might be fun to start with some of the basics and the history with some of the early modeling, so that there's a lot more to look into that if you were interested, but I'm glad to see that you like that. That's cool.
Yeah,
There's also a lot of other types of models out there. This was sort of a very biased version of what I showed you, because there's lots and lots of other good muscle models or cross-bridge models over the years that have been very, very useful. And that's, I think, important, that I think most of us older people know. But one thing to remember in your training is, there's a lot of very useful things coming out of computational modeling, and a model is very specific to the questions you're trying to ask, and it's a very unique hypothesis, your model, but it's never really accurately correct. There's a lot of stuff that's always going to be missing just because there's a lot of stuff we don't understand. But trying to keep going between the experiments and the modeling has always been a really helpful way for me to learn about how things might work, about physiology. And I think there's a lot of really, really good people that are on this phone call or on this Zoom that feel the same way and have a lot of experimental knowledge and a lot of computational knowledge that we're probably excited to build up and share with each other. So that's what's very cool about the center. So good job, everybody.
Thanks so much for a really clear introduction. I think we have a lot of new students who are completely new to the field, so having that historical background is definitely helpful, as well as the background reading. So thank you for that.
Yeah, and I just want to, I think so far of the 3 workshops we've had, this has been the most interactive from the point of view of trainee participation. So I think, kudos to you, Bert, for making it accessible, and thanks to all the trainees for participating in the discussion. And we really hope that that's going to be an increasing trend as we go through the remaining 5 workshops. So I just love your contributions, and this was an outstanding discussion.
Yeah, thanks a lot, you guys. I really appreciate the discussion. It's nice to talk about these ideas.
All right. I guess we're ready to sign off. Thanks so much. And as usual, we will post the recording in the folder of workshop materials. Good to see you all in your...
Before we sign off, I just want to say that the same QR code that was posted in the general channel will be, you just type in the name of the presentation. So I'll be sure to just send out a reminder of the QR code so everyone can find it relatively easily.
Hey.
Yeah.
Thanks, Bert, for your presentation. That was really nice to see. And we're trying to make some models that will be faster for mechanical simulation. But you should know Tim Vanderzee is working with us, looking at like as a Halec model as well to go even faster. And and comparing it with the cross-bridge, like a 2-filament model.
No, that's great.
Well,
We also have a data set from Skin Fibers that was published, the porcelain data set that might be of interest to other people as well. Kisa, you might be also, because we do all these stretch shortened cycles that of different amplitudes that change the short range stiffness.
cool. We look forward to
We'll
learning
get
more.
it together at some point and get it out, but
Yeah.
Cool.
Right now, we're really looking to data mine data for this system ID stuff that we're doing. So yeah, I'll maybe be pestering you about if I have trouble finding the data.
no, right now it's just the summary data are online, but we're fitting the time traces, but it's a good sort of, we don't have sinusoids and stuff, but we have a lot of carrot-shaped triangles of different sizes and multiple ones with different timing. So it gets you a full suite that is useful for testing models. And basically we can't get a model to fit all of it yet.
Yeah, that would be great for like, seeing whether, relatively simple model, 4-parameter model with, 2 stiffnesses and 2 dash plots could fit those complicated data. But I would be interested in like the time series data rather than the overall summaries.
No, absolutely. Like we have, Kyle Bloom tried to fit it with a couple springs and dampers. And then we only fit the dynamic parts. There's a later part with like force enhancement and mostly force enhancement that we also haven't fit either. So it's fairly rich in like various things that it has. But it's in a skinned fiber, skinned activated fiber. Lots of activation levels though.
Fabulous.
We still need to talk about how, like, trying to curate data sets to share across our network. And so we can apply different tools, our different modeling tools to them and share and compare our results.
Yeah.
I
think we sent some to Madhu at some point.
Yeah, but
maybe a first step is just to get a list of what data people have in their labs, and then see how much we have and how similar the data sets are, right? I think just surveying each of the groups might be good.
Yeah, that's a good point. Yeah, we should just do a survey of the groups and see what we have, make an inventory, and then we can work towards curating it together. Yeah. Okay.
Great.
Someone else going.
Any last minute comments before we say goodbye for now? Until next Tuesday, when we'll have Simon Sponberg as the presenter and a live demonstration of a work loop in Manduka Moss. I think I got that right.
I think so. All right. Good to see everybody.
Yeah.
Thanks.
Bye.